Welcome to Learnpedia, the 24/7 JEE and NEET resource at your fingertips. Now, take a look at this actual NEET question and see if you can answer it. If you think you got the answer, post your answer in the comment section below. To know the best way to answer this question, watch this video further.
Let us now discuss generation and conduction of nerve impulse. To discuss impulse conduction, we have to understand few basic things. Let us suppose this is exolemma. Fluid inside exolemma is exoplasm. Fluid outside exolemma is ECF. Both ECF and exoplasm contains two types of cation, sodium and potassium. The concentration of sodium in ECF is far more than exoplasm, I can call it ICF, while concentration of potassium is far more in exoplasm than ECF. I am not saying that sodium is altogether absent in exoplasm. Sodium is there, but the more amount of sodium is deposited in ECF. In the similar way, more potassium is deposited in ICF. Apart from that, exolemma has two main types of channels. One is sodium channel, one is potassium channel. Sodium channels are meant for influx or efflux of sodium, while potassium channels are meant for influx and efflux of potassium. If we say amount of sodium is high or very high in ECF then ICF, then we can say the membrane is in polarized stage. And if we say influx of sodium start to continue from out to in, so sodium get accumulated more inside, then such stage is called depolarized stage. At polarized stage, we can observe two things: more sodium in ECF and more potassium in ICF. During depolarization, there will be only influx of sodium and efflux of potassium will result in repolarization. So let us now discuss all these three terms like polarized stage or polarization, depolarization and repolarization in detail. So first is polarization. Let us now discuss something about polarized stage. Number one, at polarized stage, more sodium ions are present in ECF than ICF. This ratio is around 10:1. At this stage, membrane is electropositive outside and electronegative inside. Membrane has some fixed and ionic proteins at inner side. So finally we can say at polarized stage, more sodium is outside than inside. Membrane is electropositive outside and electronegative inside, and this negativity is due to some fixed anionic protein. One more thing, at the same stage, more potassium are inside than outside, and the ratio of potassium is 30:1. This is in ICF is to ECF, means exoplasm is to extracellular fluid. At this stage, potential across the membrane is -70 millivolt. One more information, at polarized stage, membrane is more permeable for potassium than sodium. Some potassium shows efflux and some sodium shows influx. Sodium potassium active exchange pump causes efflux of sodium and influx of potassium to maintain ionic equilibrium across exolemma. Here I said without any impulse, some potassium is going out. Without any disturbance or impulse, some sodium is going in. So sodium pump and the potassium pump, which work actively, will cause efflux of sodium and influx of potassium. So here I have used the word active pump, so it means energy is consumed at resting stage. So please try to notice at resting stage we are showing expenditure of some energy. So this is the special feature of the polarized stage. Why membrane do such influx and efflux of sodium and potassium respectively? So we can say such influx of sodium and efflux of potassium without any impulse or disturbance continues. This helps to maintain dynamicity of ions across exolemma. So finally, at polarized stage, we can see there is more sodium outside than inside, and there is more potassium inside than outside. Membrane is electropositive outside and electronegative inside. Inside negativity is due to some fixed anionic proteins. At this stage, potential across the membrane is -70 millivolt, and such potential is called resting potential or polarized potential. One more thing, at polarized stage, membrane is more permeable for potassium than sodium, and now we'll see in the depolarization state, membrane get more permeable for sodium. So simply we can say impulse conduction is basically based on differential permeability of membrane for ions. As I had said, at resting stage it is more permeable for potassium than sodium, and during depolarization permeability get increased for sodium. Now see the three diagrams here. Let us suppose this is exolemma. It has sodium channels and potassium channels. In red color I have shown sodium channels. In blue color I have shown potassium channels. During resting stage, more sodium ions are present outside than inside, and more potassium are present inside than outside. Inside membrane there is electronegative nature, while outside membrane there is electropositive nature. At resting stage almost all the channels are closed. Please notice I said almost all the channels are closed, while some leaky channels are still open, through which some potassium goes out and some sodium comes in. So the stage which I have represented here is polarized stage. When some stimulus strikes to the exolemma, it results in opening of sodium channels. Potassium channels are closed. Sodium channels get open, which will result in influx of sodium from channels. So accumulation of sodium inside will create positivity inside. So finally membrane at inner side get electropositive, and it can be represented now electronegative outside. Now see here, influx of sodium is depolarization, and it resulted in electropositivity inside membrane and negativity outside the membrane. If you compare this condition with the polarized stage, it get reversed. So now we can say potential across the membrane get positive. Initially it was negative, it was minus 70 millivolt. Now it will reach to the level of 30 to 40 millivolt. That will be called spike potential or action potential. Well, we just discussed some brief for depolarization, that how depolarization occurs. Now we write few points about depolarization. Well, whenever a stimulus strikes to exolemma, exolemma get excited, and that results in opening of sodium channels. And we know that concentration of sodium is more outside than inside. So there will be influx of sodium according to concentration gradient. Potassium channels are closed, means potassium will not allowed to go out. So simply it means only influx of sodium is going on. So we can say positivity is accumulating inside, which results in spike potential, means potential from minus 70 millivolt reaches to 30 or 40 millivolt. You can observe the change from minus 70 to 30 if we talk around 100 millivolt. This all because of influx of sodium. One important thing about sodium channel is that sodium channels are short-lived. Short-lived means they open for less time. It's approx 0.5 millisecond. So in this 0.5 millisecond, enough sodium shows influx that it is capable to raise potential from minus 70 millivolt to 30 millivolt or 40 millivolt. So here we observed that permeability for sodium is very high, and for potassium it's almost nil. So after attaining depolarized stage, now there will be repolarization, and repolarization is mainly result of efflux of potassium. Let us now study repolarization. Repolarization is nothing but efflux of potassium. Say, after influx of sodium ions, we have already discussed that sodium channels are short-lived, means they open for lesser time. About after 0.5 millisecond, sodium channels get closed and potassium channels get open. Opening of potassium channels will result in efflux of potassium according to concentration gradient, because we know that potassium is more inside than outside. Here you can observe that efflux of potassium means efflux of positivity, means membrane will start to become electropositive outside and electronegative inside. So if membrane again become electropositive outside and electronegative inside, then we can say potential across the membrane falls back to negative zone, say back to minus 70 millivolt. Sometimes potassium channels remains open for longer time, means that will result in more efflux of potassium, means more electropositivity outside the membrane, means potential across the membrane falls more in negative zone, somewhere around minus 90, minus 100 millivolt. We can call this condition as hyperpolarization. Let us discuss with a figure. Say this is exolemma. These are sodium channels. These are potassium channels. Say there was influx of sodium, means I am showing depolarization. Potassium is already there. All the potassium channels are closed, so potassium can't go out. So at this stage, membrane will get electropositive inside. So this stage will be depolarized. After this stage, all the sodium channels get closed and potassium channels get open. That will result in efflux of potassium. Simply we can say electropositivity is going out. That will make membrane again electronegative inside and electropositive outside. So this wave or this flow of potassium will lead to repolarization, or we can say the potential across the membrane will again come back to around minus 70 millivolt. At this depolarization, it got around 30 to 40 millivolt. Now let us try to understand the polarized, depolarized and repolarized stage with some graphical representation. Let us suppose this is the zone of minus 70 millivolt. I can say polarized side. This potential is around minus 55 to minus 60 millivolt. This is called threshold potential or threshold level. Threshold level means stimulus received by exolemma should be so much that it should be capable to open so much sodium channels and should capable to show influx of so much sodium ions that it can raise potential from this minus 70 millivolt to minus 60 millivolt. If it reaches to minus 60 millivolt, definitely it will go up to the spike level. Means now again we discuss this. Say we are at this point, the minus 70 millivolt. At this point we know that this is the polarized stage and membrane is potential permeable to potassium as well as sodium, but more permeable for potassium than sodium. So there is few influx and efflux of sodium and potassium. So slight wave will be there. The wave is going. Now at this point all of a sudden we get the stimulus and all the sodium channels get open. That results in influx of sodium, and that influx of sodium raise the potential up to this 30 to 40 millivolt. So this complete process is called depolarization. It is due to influx of sodium ions. Now at this point, all the sodium channels get closed and all of a sudden potassium channels get open. If I say all the potassium channels get open, means now there will be efflux of potassium. You can see here when efflux of potassium is going on, the membrane will come back to its potential around minus 70 millivolt, but with one problem. Now at this point, if we compare this point, to this level, both are having same potential, but at this point membrane has more sodium outside than inside, and at this point membrane has more potassium outside than inside. But at both these conditions potential is same but not the ionic distribution. So this stage is due to efflux of potassium and this is called repolarization. And sometime potassium channel remains open for longer time and potential falls below minus 70 range. If minus 70 is polarized, then below minus 70 this zone is hyperpolarized. Now one more important thing which we will discuss later in impulse conduction mode. At or during depolarization, exchange pump is inoperative. You think if exchange pump is operative during depolarization, then the pump will cause efflux of sodium immediately. But if we want to get this plus 32 plus 40 millivolt level, we have to restrict sodium inside, and if pump will work, it will cause efflux of sodium. And during repolarization, again pump is inoperative. If pump is operative during repolarization, it will cause influx of potassium. So if we want that there should be efflux of potassium through potassium channels and potassium should retain outside, then pump should be in inoperative mode. So during depolarization and repolarization, pump is not working. After reaching to this point, pump get active and it will cause efflux of sodium and influx of potassium to get ionic distribution back, because at this point we have reached to the polarized potential, but still we are waiting for correct ionic distribution. So in this graph you can see clearly, if we go up, this is depolarization, means influx of sodium. If you go down, this is efflux of potassium. In depolarization it is influx of sodium, and during repolarization it is efflux of potassium. Let us now discuss one of the important facts, that how impulse runs all along the fiber. Right now you can observe each and every zone I have shown is in polarized stage. Say if I am taking this zone, this is in polarized way. If this zone get stimulus, the permeability get changed and it will become like this, that sodium will show influx and membrane will get electropositive inside. Well, I have shown both the condition, the polarized stage and the depolarized stage. This polarized stage becomes this depolarized. So whenever this zone get depolarized, I can say membrane will behave electropositive inside and electronegative outside. Now you can see with the neighbor, the positive and negative terminal develops, and we know that current always moves from positive to negative zone. So we can say this was the zone which received the stimulus externally and become depolarized, means electropositive inside, and neighborhood which is still electronegative inside. Now there will be a small cell. This is the positive electrode, this is the negative electrode, and current will move from positive to negative. Say wave of depolarization will run to the next zone, and now this next zone will show influx of sodium, means it get electropositive inside and negative outside. So here you can observe formation of one more small cell. Now current will again move from positive to negative. Same will happen with this potassium inside, sodium shows influx, zone get positive, negative outside, one more cell. So finally from positive terminal to negative terminal, from positive to negative, current will flow. Now at the same point, if we are saying at depolarized side, the inner zone is the electropositive and neighborhood is electronegative, at the same time you can observe here, this is the electropositive and this is electronegative. So if current flows in this way, current can also flows in this way. So simply we can say in an axon part, impulse conduction is bidirectional and current always move from positive electrode to negative electrode. This bidirectional conduction will become unidirectional at synapse. So we can say synapse, mainly chemical synapse, will act as unidirectional valve. So this is something about impulse conduction. Let us now discuss impulse conduction again with few more diagrams. Now see here, this is neuron and this is axon part of neuron. You can see here we have enlarged this zone and now we are enlarging this part. This is exolemma. Fluid inside the exolemma is cytosol, means it is exoplasm, and the fluid outside the cell or this neuron or nerve fiber is ECF, means extracellular fluid. We have already discussed that in ECF, number of sodium ions are in abundance, they are in 10:1 ratio, and in cytosol part, means exoplasm, the potassium ions are in abundance in 30:1 ratio. You can see here, these are sodium channels, these are potassium channels, sodium channels, potassium channels. These are not in alternate manner, they are just shown in the way. At this stage you can observe negativity inside and membrane outside will be positive, and this electropositivity outside and electronegativity inside results in a potential across the membrane is minus 70 millivolt. That is resting potential. You can see in this graph right now, this blue arrow is at minus 70 millivolt. So this is at resting potential. Now you can see here, whenever a threshold stimulus strikes to the exolemma, permeability of membrane will start to change. That results in more accumulation of sodium ions inside, means electronegativity inside will start to decrease due to accumulation of sodium ions, and potential will start to shoot up from minus 70 to minus 60 millivolt. That is now threshold level. You can see in this figure, sodium channels get open and sodium ions are showing influx. This influx will result in more and more accumulation of sodium ion inside. At this point, potassium channels are still closed, means potassium ions can't move out. So overall, positivity in exoplasm start to increase. That will make membrane electropositive inside and electronegative outside, and now potential across the membrane get revert. You can see in this graph, potential is rising from minus 70 to minus 60, its threshold level. Still influx of sodium is going on and potential will reach up to 30 millivolt. That is now called spike potential or action potential. As we know that sodium channels are short-lived, means they open for lesser time, around 0.5 millisecond. After that, potassium channels get open. Now you can see here, sodium channels are closed, potassium get open, that results in efflux of potassium. Now I can say efflux of positive ions in terms of potassium, and that will make membrane electropositive outside and electronegative inside. More and more efflux of potassium outside will result in fall in potential back to the resting potential, means to minus 70 millivolt. You can see here the graph start to fall from plus 30 millivolt to downwards. See here, more and more efflux of potassium results in fall of potential across the membrane, and finally potential will reach to this zone, the minus 70 millivolt. We have already discussed that if potassium channels remains open for longer time, then potential across the membrane may fall beyond minus 70 millivolt. It could be minus 80, minus 90, minus 100, and this point is called hyperpolarization. So you can see here, return to normal permeability, return to normal potential. But there is one problem here, and the problem is what? At this point, membrane has more sodium outside and more potassium inside, while at this point, membrane has sodium inside and potassium outside. So after attaining this repolarization potential, means resting potential, sodium potassium active exchange pump causes efflux of sodium and influx of potassium in 3:2 ratio to regain ionic distribution. So this is impulse conduction with this graphical explanation. Now you can see here in the graph resting potential, threshold level, spike potential. It's all because of influx of sodium, that is depolarization. Now from this point, fall in potential due to efflux of potassium, that is repolarization. And here we'll reach to the level of minus 70 millivolt. But the ionic distribution is not okay here because at this point potassium lies outside and sodium lies inside. From this point onwards, sodium potassium active exchange pump will work and will try to regain the ionic distribution, which will come here at this point. Now in this graph we can observe two more words. One is absolute, one is relative. What are these? These are refractory period. Refractory period means it's a time gap between a stimulus received by a nerve fiber, or it's a time gap between response of a nerve fiber to one stimulus till it get capable to respond to the next stimulus. So we can say during depolarization and up to repolarization, membrane will not respond to the next stimuli. So we can say it is absolute refractory period, while from this point onward, due to some efflux of sodium and influx of potassium, membrane again can get some stimulus. So we can say this is called relative refractory period. So when membrane is not receiving any stimulus unless it comes to this point, then it is called absolute refractory period. If it is capable to get some stimulus, then it is called relative refractory period. Now after getting this point, again membrane is capable to show this flow, means for next stimulus, the depolarization and the repolarization.
Hey there, hope you understood the concept. Here's the solution to the question asked at the beginning. Hit the bell icon below to subscribe to the Learnpedia channel and to learn more through such interesting videos. Learnpedia's JEE and NEET prep tools contain more than 4,000 videos and over 20,000 questions. These can be accessed online through our website or offline through an SD card or a pen drive. To buy now, visit www.learnpedia.in. You can also try a free demo of our product before buying.